Hello everyone. We will study mycology from the book edited by Tariq Mohammed Abdel Ghani and Hussein Hosni El Sheikh. So it's a very nice book and we are going to study the whole mycology from this book. So let's begin. So first of all, what is mycology? Mycology, it is a branch of biology which deals with the study of the fungi. Okay, and their genetic and biochemical properties, their taxonomy, their use, their importance to humans as a source of medicine, food and psychotropic substances. Also they are consumed for religious purposes as well as they are dangerous as well, such as poisoning or infection, destruction of food, lumber, paper, cloth, toxins produced by poisonous mushrooms, and within food mycetism and mycotoxicosis, and they are compared with other science as a relatively new science that becomes systematic after the development of microscope. Okay, so I'm not going much into detail about this. Let's start with the description. So what are fungi? In singular we call it as fungus. Okay, plural is fungi. They are multicellular with some exception like yeast Saccharomyces cerevisiae, which is having prominent unicellular fungus. They are heterotrophic, means they are deriving their energy from another organism. They can be live or dead, and they are having some cells with two nuclei or more than two nuclei, multinucleate or uninucleate per cell. So they are widely distributed in nature, be it be soil, air, or water, some organic debris, and there are around several types of fungi. There are groups of organism, that is fungi, and they have transferred out of the kingdom fungi in part because of fundamental biochemical differences in the composition of the cell wall. Okay, so they have been given the separate kingdom because their composition of the cell wall is different from another organism. Okay, and some of the true fungi, they have a cell wall which consists of chitin and other polysaccharides. Okay, their cell wall is made up of chitin and other polysaccharides. True fungi do not have cellulose in their cell walls, but some fungus like organisms do. Okay, they are, their cell wall is made up of chitin, but some fungus like organisms have cellulose as well. So let's study about true fungi. What are true fungi? So not all the species have cell walls, but in those that do, there are three layers of cell wall. Okay, which follows the plasma membrane. So from inside out, what is the first layer? If this is a fungal cell, if this is a plasma membrane, inside out, what are the layers? Okay, so outermost is the chitin layer consisting of unbranched chains of beta 1,4 linked N-acetyl glucosamine. Okay, in short, we will say as GLN, glucosamine. Okay, which is present in Ascomycota and Basidiomycota, or poly beta 1,4 N linked N-acetyl glucosamine, which is chitosan, in Zygomycota. Okay, so in Ascomycota and Basidiomycota, there is beta 1,4 linked N-acetyl glucosamine. Another layer, beta 1,3 glucan, beta glucans, which are glucose molecules linked via beta 1,3 or beta 1,6 bonds. Okay, so these beta 1,3 glucan, these are kind of glucose molecules which are linked by beta 1,3 or beta 1,6 bonds, and these provides the rigidity to the cell wall. Okay, while what do alpha glucans do? Alpha glucans are defined by alpha 1,3 or alpha 1,4 bonds and function as a part of the matrix. Okay, not the wall but the matrix. Okay, and the third layer is manoprotein, which is mannose containing glycoprotein. So most of the proteins found in cell wall are glycosylated and they contain mannose. Thus, these proteins are called manoproteins or manamins. So, true fungi do not have cellulose in their cell walls. They have N-acetyl glucosamine, okay, which is beta 1,4 linked. Another layer is, so this is number 1, this is number 2, okay, and the inside is number 3, which is the manocell layer, okay. Moving forward, fungus like protist. Okay, so oomycetes, they are group of saprophytic plant pathogens. Saprophytic plant pathogens means feeding on dead. Okay, which also known as water molds. So, what is the another name of water molds? It is oomycetes. What is the another name of oomycetes? It is water mold. Okay, and previously they were told to be as fungi, but there are some evidences which led to their reclassification as heteroconts. So these heteroconts, they are related to a brown algae and diatoms. Okay, so fungus have a typical cell wall made up of chitin, but oomycetes, they have the cell wall made of cellulose and glucans rather than chitin. Some genera like Acyla and Saprolegnia, they do have chitin in their cell wall. So oomycetes, some examples you will find like Aclea and Saprolegnia, they have chitin in their cell wall instead of cellulose, but they belong to oomycetes. Okay, and the fraction of cellulose in their walls is more than 4 to 20%, which is far less than comprised by glucan. So more amount of cellulose is there instead of glucan. Also, one additional thing, that they have hydroxyproline amino acid, which is not usually present in fungal cell wall, but these have. So they have hydroxyproline in their cell wall, which is not present in fungal cell wall. Okay, so what peculiar characteristic we understood about oomycetes? One is amino acid. Okay, another is cell wall, which is made up of cellulose. Till now we have found those. And next is the dictyostelids are another group of formerly classified among the fungi. They are slime molds. Okay, so one category is oomycetes, another category is dictyostelids. Okay, oomycetes are known as water molds, and these are known as what? These are known as slime molds. Oomycetes have a characteristic that they have hydroxyproline. And what about dictyostelids? So these slime molds, they feed on unicellular amoebae. Okay, they aggregate in a reproductive stalk and sporangium under certain conditions, like when the condition is adverse in nature, they form a stalk and the sporangium to kind of hibernate. And the cells of this reproductive stalk as well as the spores which are formed at the apex, they pose as a cell wall. So even dictyostelids have a cellulose cell wall. Oomycetes also have a cellulose cell wall, and the spore wall has shown to possess three layers. Okay, the spore walls, which is formed in adverse condition, this spore wall is formed of three layers. Okay, the spore wall is formed of three layers. Okay, and this is composed primarily of cellulose, and the innermost is most sensitive. The innermost is most sensitive to enzymes of cellulase and pronase. Okay, the innermost region, it is sensitive to cellulase, which degrade the cell wall, and pronase, which is a mixture of protease. So we studied about the fungus like protist. Fungus like protists are what? They are oomycetes and dictyostelids. Oomycetes also have cellulosic cell wall, and they have hydroxyproline as well. What about this Dictyostelids. So dictyostelids, they are another group and they have cellulosic cell wall. They form sporangium under the adverse conditions, and the sporangium have three layers made up of cellulose, and the innermost layer is most sensitive to some enzymes like cellulase and pronase. Okay, so this is up to where we studied. Next we are going to study about the general characteristics of true fungi. So general characteristics that fungi are eukaryotic. That means eukaryotic, any cell which is eukaryotic have a membrane bound nuclei containing chromosomes and some cytoplasmic organelles within them. So they are filamentous. Filamentous means they have some hyphae like structures which has apical growth and which branches to form a network of hyphae. So one hyphae will form a network of hyphae, and the network of hyphae is known as mycelium. Network of hyphae is known as mycelium, and there are some fungi which are unicellular as well. Example, yeast. This unicellular cell have all the organelles. So protoplasm of hyphae or a rigid wall surrounds cell of a fungi. Protoplasm is present. So this consists of glucans and chitin. Please remember it consists of glucans and chitin. Although the cellulose enters the walls, which has the composition like which is the composition of some of the fungi, and many fungi reproduces both sexually and asexually. Okay, so spores are produced as a result of sexual and asexual reproduction in most cases. We'll go through it in detail in following some examples. So fungal nuclei, they are typically haploid, and hyphal compartments are often multinucleate. So if this is a hyphae and there are several compartments, it can be multinucleate also, more than two or two. Okay, so diploid nuclei present in Mycota and some yeast. So majorly are haploid, but diploid nuclei are present also where in Mycota and some yeast. Okay, and how they take the nutrition, they are chemo heterotrophic, that is chemo organotrophic. They take their sources of carbon from environment and through biochemical reactions, and storage compound in the form of lipids glycogen like humans and sugar alcohols, and they can be free living or they can form a symbiotic relationship or a parasitic relationship or with other organisms or maybe plant. So how does a fungus feed? So fungi absorb food from their surroundings, okay, depending upon what type of fungi it is and what type of surrounding they are living in. So they grow generally in stringy strands called hyphae. Hyphae are kind of thread-like structures which absorb nutrients and water from the surrounding and give energy to the growing fungi. Okay, and this network of fungal hyphae, like hyphae in clusters, network of fungal hyphae, it is known as mycelium. Generally they aggregate and form the network known as fungal mycelium. So this term is very common, hyphae and mycelium. You will find it repeatedly. I'll be repeating this. So please remember hyphae and mycelium. Okay, so fungi grow on like a stringy strands called hyphae. These hyphae form a network known as mycelium. Many hyphae grows together, form a network known as mycelium. So their nutrition is chemoheterotrophic, which is chemoorganotropic, that is synthesizing the organic compounds they need for growth and energy from some pre-existing sources. Okay, using for chemical reaction, and the protoplasm is protected by a wall which is chitin in most of the cases, and nutrient is absorbed by the process of absorption. So what are the raw materials they use for? They use simple sugars, amino acids, which is present in the medium. They can absorb it directly from the fungal wall and plasma membrane. Okay, and what about complex molecules? They break down complex molecules like polysaccharide and proteins. They are broken down into smaller molecules and then they are absorbed. So how they are broken into smaller molecules? By some enzymes uh, Degradation is achieved by some enzymes which is released through or bound to fungal wall. These enzymes are either released or which are already bound to fungal walls, which act on the outside of the cell on the some larger molecules to form simpler molecules, and they are known as extracellular enzymes. Okay, so what are the types of fungi? They are saprophytic, they can be parasitic, they can be symbiotic. So saprophytic fungi, as the name says, they feed on dead materials of plants or animals, or on some waste materials such as dung. Okay, and they are either obligate or facultative. Obligate means compulsorily, facultative means changing according to the situation. They are found in woodlands where the floors are covered with massive amounts of dead plant material such as fallen leaves, twigs and logs, and many mushrooms feed in this way. Okay, so second type is parasitic fungi. What are parasitic fungi? These fungi do not wait until the plant or animal is dead. Okay, unlike saprophytic, they feed on it when the plant or animal is living. Okay, so they feed when the organism, when their host is still alive, so they can cause serious damage to the organism on which it is feeding, and they can grow inside humans or other animals and cause some diseases like athlete's foot, ringworm. Okay, you have heard of the Betex cream which is used to treat the ringworms, and they can be obligate or facultative. So they can be also obligative and facultative, they are also obligative and facultative. The third type is symbiotic fungi, which you must have heard of, that they live in a mutual condition, mutual relationship with other organism. They live inside or on the living beings, and they do not cause the damage when they are in symbiotic relationship. Okay, so they both, the host and the fungus, benefits from each other. They live in a harmonious relationship that some will give and some will take. Means like both will give and take. Okay, so say for example, they live in symbiotic relationship with many orchids, and here the fungi grow underground, and their thread-like hyphae grow into a thick mat known as mycelium. So since they can grow in a thread-like manner and form a mycelium, which this mycelium, when they grow near the root area, it increases the surface area of the root. Okay, and when the surface area of root increases, they can absorb more nutrient and more water when alone, more than the root hair can do it alone. Okay, and they passes this nutrient and water to the plant, okay, through the roots of the tree, and in turn plant provides the prepared food to the fungi. So this, the relationship between the fungi and trees, they are known as mycorrhiza. Okay, another relationship is lichen, where the relationship is between the alga and the fungus, where this grows as a, and the lichen looks like flat green or yellow growth. You will find colored substances onto the soil or onto the plant bark. Okay, so on the rocks and the trees, and this lichen is a fungus and an alga association when they are growing together, helping each other to survive and grow. Okay, the fungus is providing the shelter, and alga part is providing the food. Okay, next is some general cell biology of fungi. So they are described to be grown as threads known as hyphae, singular is hypha, and these hypha can be branched and they can be dense mass called mycelium. You can follow the structure. A hypha develops from a spore, and it can be this can be branched, or when they form in network, they are known as mycelium. Okay, this is mycelium. Okay, this is branched hyphae. Okay, so hyphae develops from a spore, and they are surrounded by rigid chitin containing cell wall. Okay, the hyphae extends by tip growth and multiplies by branching, creating a fine network known as mycelium. Hyphae contain all organs including nuclei. Mitochondria means a typical composition of the eukaryotic cell, and which is first bound by plasma membrane, and outside to the plasma membrane is the cell wall, and there are microtubules and endoplasmic reticulum also present. And hyphae is essentially a tube like structure consisting of rigid wall and containing protoplasm tapered at its tip. Okay, this is the region of active growth. So the upper part is the region for the active growth, which is the extension zone. So these hyphae can be septate, which have cross walls. It can be septate, which can have cross walls. And if septa is present, so they can be septate or aseptate. Okay, if present, or septa can not be present. Okay, aseptate and septate. Okay, so when it is observed under light microscope, it was found that some fungi possess septa, which divides the hyphae into separate cells, and it as you have studied that some Mycota have two nucleate also. So some septa possess one or more pores, such septa divide the hyphae into a series of interconnected hyphal compartments. Okay, so when the pores are present, it will divide the hyphae into several compartments. Okay, rather than separate discrete cells. Okay, so it can be like this, where the separate cells are not present, but several compartments are there. Okay, several compartments are there rather than discrete cells, and this xenocytic hyphae lack septa. Xenocytic means where there is one cell only, numerous pores are there, but they are multinucleate. Okay, this is kind of xenocytic structure. So the cross walls form only to isolate old or damaged regions of a hypha, or to isolate reproductive structures, either to separate the reproductive structures from vegetative structures, or to isolate the old from or damaged regions from the new ones. Okay, understandable. I hope it is understandable to all of you. These we are doing only the basic part, so that we will be aware of the terminologies, and when we will go into deep into the description and detailment of each of the genus, then it will be more clearer to you. So you can see the mycelium is a network of hyphae. This is a septate hyphae. Septate hyphae have separate cells, pores, nuclei. This can be haploid. Majorly, it is haploid. In some cases, it is diploid as well. There's xenocytic hyphae where the pores are present, but the nucleus is present. This is a single cell where the nucleus are present in a several nucleus are present in a single cell. This is known as xenocytic hypha. Okay, understandable. These are the different forms of hyphae, septate hypha, and this is kind of a septate hypha or xenocytic hypha where several nucleus are present in the xenocytic. So the plasma membrane is closely associated with the hyphal wall, and in some regions even be firmly attached to it, making it difficult to plasmolyze the hyphae. Okay, so plasma membrane is associated with the hyphal wall, which makes it difficult for the hyphae to plasmolyze. Okay, so again, as it was said earlier, there can be the presence of one or more nuclei, and the septa poses large central pore in some species, and the number of nuclei within the hyphal compartment will not remain static, because the nuclei are able to pass between adjacent compartments via the pore. So if there is a pore present in the hyphae, the nuclei will not remain static in one position, it will remain moved to another position. Okay, so other cytoplasmic organelles are found, which are nuclei. So as we all know, the description is kind of similar. So fungal DNA, there is a less complexity than other eukaryotes. There are fewer repeated DNA segments compared to other eukaryotes. Uh, then nuclear envelope does not break down during division. Okay, in case of other eukaryotes, nuclear envelope breaks down during division, but here it does not happen. Chromosomes are not distinct. These are well known characteristics we need to know. Then mitochondria elongate with plate like cristae. Endoplasmic reticulum are a little narrow bound channels. Golgi are ringed cisternae. They are not flattened sacs, they are ringed cisternae. Okay, so the growing tip is structurally and functionally very different from the rest of the hypha. Okay, from the rest of the hypha, the growing tip will be structurally different. Its cytoplasm appears denser. How the growing tip will look different, as we have studied about the meristematic tissues also while reading the cell division of the stem or the root. We have found that the cells which are in active division, their cytoplasm appear denser, and there are no major organelles at the extreme tip. At the extreme tip there is an accumulation of membrane bound vesicles, which is known as apical vesicular cluster (AVC), which plays an important role in apical growth. And vacuoles may also be visible, and they are smaller at first, then they grow larger and merge with one another, and they restore some cellular metabolites like enzymes and nutrients. So, in the oldest part of the hyphae, the protoplasm may break down completely. Okay, to distinguish the older cell from the newer cell, the protoplasm breaks down completely due to their autolysis or self digestion, or in natural environment heterolysis. What is heterolysis? It is a degradation due to the activity of microorganisms. Okay, self digestion is like breaking down on its own. Heterolysis is with the activity of another organism, like activity of another microorganism. So let's understand a little about the ultrastructure of the hyphae. So septum is present here. There are some lipid bodies, some crystals present. Nucleus is very simple, where the nuclear envelope does not break down when there is cell division. This is hyphal wall. Plasma membrane followed by chitinous cell wall. Vesicles are present at the tip part, then cytoplasm is present at tip wall. Plasma membrane is there, mitochondrion is plate like cristae, ribosomes, endoplasmic reticulum is quite narrow, vacuoles become larger later on, which helps in apical development. Okay, so that's it. We will proceed with fungal growth in the next video. Thank you.